This is a production of Cornell University.
Okay. Well, first of all, thank you so much, Ginny, and also to Solveig for inviting me to be here, showing me around. Um, I'm just hugely excited to get to talk to this particular group. Ginny said lots of familiar faces, also many that aren't. So all I will tell you about like my background. Hmm, uh, okay, right. So the reason that I was here from 2013 to 2022. Actually, I was here for about 6 years, then moved remote. But I worked on the next generation cassava breeding program, was hired by Jean-Luc in 2013. And that was really the time of my life. Well, it was the time of my life. And I fell in love with plant breeding. And um, so I am doing I suppose I was working on food security, providing decision support to plant breeders in the cassava breeding project mainly during that time. And what I've done now is shift gears a little bit, trying to run my own breeding program. And my focus is on cover crops and maybe breeding for sustainability aspects in our cropping systems. So I am at Auburn University now. Auburn is all the way down here. We're kind of west of Atlanta. We're in central eastern Alabama. We're the Land Grant Institute for Alabama. So I was appointed as assistant professor of quantitative genetics, which means I can basically work on whichever cropping systems that are fundable and that are supported by stakeholders. And there's a lot of motivation and work around cover cropping as well as forages in Alabama. So I've set up myself as the Auburn University's cover crop and forage breeding lab and trying to support Southeastern growers. But the research focus that I kind of have taken on is breeding for intercropping, and that's what I'm mainly going to talk about today. So I have really been starting a breeding program from scratch at Auburn. There were several successful forage breeders in previous years at Auburn University that have developed especially forage legume varieties and germplasm. So my kind of first job was, but the last one had retired in 2016. So my kind of first job was exploring the coldrum and trying to figure out what kind of germplasm we have lying around and what will actually grow. So one of the cool things taking on cover crops and forages is I get to work on probably too many different species. However, I'm going to focus today and just talk to you about my sort of primary species that I'm loving to work with, which is crimson clover. This is Trifolium incarnatum. It's not super common around this region, but it's well, it is super common where I sit. And we've had several releases of crimson clover from Auburn University in the past, so we had germplasm to get started with. It's actually like the lead cover crop legume in the U.S., at least according to like SARE cover crop surveys. Importantly, and I honestly didn't appreciate this fact until I took on work in this area, it's a cool season legume and it's allogamous, which means we're breeding an open pollinated species here. We do know there's some self-compatibility in crimson clover, but basically those are my problems now to figure out how to navigate. We don't really have genomic resources in crimson clover. There's one study published in the past with AFLP markers in the species. So we're working, I'm not going to talk much about it today, but one of the things we are working to do, build a crimson clover reference genome, do some resequencing, try to understand the population genetics in this species. Now, before I go into like the actual work we're doing, I'd say the most tangible, real thing that I do and I have done in the last couple of years is grow a team that work with me. We're currently 7. I have two PhD students working with me, 2 master's students, and also an undergrad, Anthony, who's actually going to start a master's degree in just a couple of months, and a postdoc, Cynthia Gogo, who started in September of last year. So I am really fortunate to get to work with these folks. Several of them actually came out of the cassava breeding and genetics world, so we kind of speak the same language, which is cool. But we're a little bit of a diverse group. Also, just to say before I go on, starting a breeding program and learning from the angle I came at all of this was not as a plant breeder. maybe more of a molecular breeder, let's say. So starting a breeding program that includes field work and that kind of thing takes a lot of help. I have to ask a lot of questions and depend on a lot of people. So I'm not going to try to name names, but I'm very fortunate to work with amazing people, including the Cover Crop Breeding Network, which I'll mention in just a moment. Starting breeding program also takes funding, and I've been fortunate in the last few years. We have an Alabama Ag Experimental Fund that's helped us pilot some of this work. We've actually gotten some funds from Alabama Cotton Commission and the Wheat and Feed Grains Checkoff, so they're supporting cover crop breeding activities, at least so far. We have 2 USDA NIFA projects that are supporting the intercropping work I'm going to talk about today. JGI is helping us develop clover genomic resources, and 2 of my grad students have SARE grad student grants. So that's been really tremendous to help us get started. So, and importantly, talking about needing a lot of help, I've partnered up with the Cover Crop Breeding Network, who I more or less assume this audience knows about this organization, but it's led by very own Ginny Moore, coordinated by Solveig Hansen. A great community, sort of putting me with a group like I was working with Next Gen Cassava, I would say. And actually, so I started by just planting, a rep of an advanced line field trial 2 years ago, and they actually asked me to be the Crimson Clover breeding lead for the network. And that puts me into a position I'm actually a little more familiar with. I provide decision support to the various breeders in the project. I haven't mentioned, we're a collaborative of plant breeders, agronomists, soil scientists. We're all collectively working to develop improved varieties for cover cropping. We have 3 legume species that we focus on: winter pea, hairy vetch, and my favorite, crimson clover. We also have a cereal rye breeding program, and we have a winter canola program as well. So each species has 1 or more different breeding nurseries at different sites across the nation, with breeders focused on those particular populations. So my role is really to sort of crunch the data and provide decision support when they need to make culling or advancement decisions, as well as I am now doing that for my own Crimson Clover breeding program, which has been really great. In case I forget, or I shouldn't forget to actually mention, cover crops are those that we're planting to benefit the soil and the subsequent crop, rather than for harvesting. So these are crops that we're growing to benefit the soil. They have a myriad of different potential benefits when you optimize them, depending on what your goals are. So usually I classify the benefits of cover cropping as services, sometimes called ecosystem services: suppressing weeds, scavenging nitrogen, helping to prevent erosion, those kinds of things. There at least have been in the past economic incentives that also support farmers to plant cover crops. The benefits of cover crops are, some of them are, you can see them immediately, right? Like having a cover crop residue can prevent a washout when you have heavy rains in the early season, and many of them you have to practice for a long time to build up soil organic carbon or improve the aggregate of your soil. So long and short-term benefits aside, those are the reasons that, cover cropped acres are increasing year on year nationally. So across most of the commodity groups, down where I sit in Alabama, we're actually have a pretty, we're higher than the national average for cover cropped acres. And there are a number of other researchers who are mostly in the area of agronomy, but also variety testing, are working on cover cropping. So in the CCBN, each species has some of its own breeding goals, and I'm not going to go into those in detail. In general, we're selecting for high biomass and vigor. High biomass, the amount of cover crop residue, is the chief thing that we can use to evaluate how much of those benefits the soil may actually get. So the thing that I want to add to this, at least today, is the need to select for performance in mixtures. This is a simple pie chart from Cover Crop Survey that shows that 65% of farmers who cover crop plant mixtures when they do. So usually that's grass legume mixtures, but sometimes they're planting like up to 15 species or huge numbers of species together. So we need to select in these environments. Okay, so finally, an outline. So I'm going to talk to you first some conceptually about breeding for intercropping, why to do it, how to do it, and then try to talk to you about actual efforts that I've been making to do this. So I will spend most of my time talking about a cover crop mixture project, which is focused on crimson clover and oat, but I'm going to mention briefly a small project I have with cassava and cowpea, and then last but definitely not least, a project I have with Jean-Luc and others who may be familiar to you that's focused on grain intercropping with oats and peas. All right, so um, so most of what I'll talk about came from two papers we published in the last couple of years, a perspective article I wrote with Jean-Luc, Mikey Cantar, and Nick Santantonio, and also a review paper that Ginny led about plant breeding for temperate intercropping systems. So intercropping is anytime we're planting two or more species and or cultivars in the same place and or time. So that's my very, it's a big umbrella term for a very broad practice. There may be as many intercropping practices as there are people who plant intercrops. They can be categorized different kinds of ways, basically based on how the crops that we're planting are spatially or temporally segregated. I'm not going to give too many examples, just to say that my focus has been full mixtures, meaning the pair of species we're talking about are mixed in the same hopper, planted in the same furrow, they're growing in the same row, and they're going to interact with each other for better or worse for the whole time. So why intercrop? That could be a whole talk, and again, it depends on the intercrop. I'm going to be very reductive here and give you basically two kinds of reasons that intercropping can be beneficial. The first one is, like we like to see, over-yielding, meaning that if we plant a mixture on an acre rather than the two species in monoculture, 2.5 acres say, we get a better result, higher biomass, higher economic value of the intercrop. That's, of course, the ideal that we're going for, right? That's not what always happens, but that's the reason to try to do the practice. And the other kind of reason is something we often refer to a little more loosely, multifunctionality. And I'll just be simple about this and say it means more species, more features, right? So for example, cereal rye, a grass, can help with nitrogen retention, but mainly it's going to produce a lot of biomass, add a lot of potential carbon to the soil. Pea is a legume, it's going to fix nitrogen, add some N that wasn't previously there. So what we're meaning here is if you have rye and pea, you can get some of the best of both of those worlds, right? Okay, so there's a bunch of steps we might go through to set up a program to breed for intercropping. This is a figure from our review paper a couple years ago. I'm going to kind of simplify this down rather than walk through it. So first of all, we need some enabling factors, and I'm just going to kind of say like the motivations and the funding and the product profiles we need to target are there already. So let's assume that and think like geneticists and breeders. So firstly, we need to like actually make sure there's genetic variation for performance in intercropping. If there isn't in our germplasm, we're doing a management. We need to be focused on agronomy, right? Assuming there is, then we're going to have to ask where the variation is actually expressed. In other words, genotype by management interaction. Because if, like it shows here, there's no interaction effect, so varieties are not re-ranking at all between environments, you can just keep your breeding program as is, right? Select for the better of whatever it is, and you'll get something that's better for intercropping. But we're typically going to find that intercropping is a different environment than monocrop. So if we have more simple like non-crossover interactions, I would say we don't have rank changes. You can do your breeding for intercropping under monoculture conditions, which is what we're of course going to want to do, right? Plant breeding's hard enough. We've already got our breeding programs operational, presumably. So when we're usually selecting under monoculture for monoculture. So if we can avoid adding brand new field trials or instituting a new pipeline, that's what I think most of us are going to want to do. I do want to say, like at this point, if we're going to keep selection under monocropping, we can reach for indirect selection, meaning we sometimes call this the trait-informed approach. Try to identify phenotypes that are expressible in monocrop but predictive of performance in intercrop and select for those. But I'm really going to focus today on the situation I think is going to be very common. We have crossover interaction, right? The things that are best for monoculture are not those that are best for intercrop. When that's the case, you're going to have to institute some co-evaluation. You're going to have to breed for intercropping in the actual environment where you have intercropping. So this is my very simple continuum of possible co-testing strategies, with the simplest being what we'll call a tester scheme, right? We're going to keep the germplasm primarily separate. We're going to take all of species A and evaluate them with a few exemplars of species B. We're going to do the reciprocal, all of species B, with a few exemplars of species A. We'll call them testers. The opposite end of that, we're going to collide two entire germplasms as early as we can in a breeding program. We're going to have some kind of a novel breeding pipeline that does that. That's going to be a brand new thing and potentially a lot of work. So we are not always going to do that kind of joint evaluation, but what I want to talk about is how we can actually do that with some of the modern approaches that we use in plant breeding. So I actually don't have to invent a lot to get this started. Strategies to breed for intercropping have been around. This is, I think, the first paper that describes it. It's 1985 by A.G. Wright. And already, hopefully, you can see that this might resemble like a hybrid breeding program where you have two heterotic groups, but instead we've got two species. Obviously, we're not crossing species with each other, but what this is meant to indicate is we're going to have a field trial where we'll assess members of both species co-planted. We will then use those data to make rankings, breeding values, and make selections either in species A and or in species B to generate new progenies. And then we're going to have to, of course, field test them when intercropped. This paper also described how we can genetically evaluate an intercrop. So there are some different models than what I'll show here, but this is basically, we call it a general and specific mixing ability model. It has direct analogy to combining ability model. We would use in hybrid breeding. So here we're going to evaluate a response variable is going to be the total merit of the intercrop, the combined yield from your plots. So like for cover crops, that's just going to be the total biomass, for example. For others, it might be like an economic merit index or whatever you might be focused on. So basically in this model, we have two main effects, one for each species. We like to call this a general mixing ability. That's the average performance of a genotype in mixture with any other genotype of the alternatives of the companion species. And we have a specific mixing ability term, an interaction term. That's the performance deviation of particular combinations of the two species, of cultivars of the two species. So we have the statistical or the genetic models that we need. We have some idea of what the breeding strategy might look like, but the challenge to do this is we're talking about breeding in genetics with more than one species. One species is generally enough for most of us. There's a huge number of possible combinations and interactions, like genotype by species, by management, by environment interaction. It's a lot to navigate and parse. You just take the genetics, like the genetic component. It's quite easy to imagine 200 lines each per cohort for any given pair of breeding programs. That would be 40,000 combinations, which we're never going to try to do a field experiment and co-evaluate all those combos, right? Certainly not replicated yield trials or anything like that. So as you might imagine, that limits what we can do in a conventional kind of phenotypes only selection paradigm, right? We're going to have to, in that case, evaluate all or at least most of the combinations that we want to have a ranking for. So we end up limited to the late stages where we have a low amount of genetic variation. So as always, we want to reach for approaches that allow us to evaluate intercrop performance or whatever the trait is we're interested in as early in a breeding pipeline as we can, so that we can enrich the downstream results for higher performance. In other words, to increase selection intensity. And so we can reach for basically two components that are fairly common these days. Firstly, we can use a sparse design. So we're using these commonly to capture genotype by environment interaction without having to replicate all of your genotypes multiple times in each environment across every single environment. For intercrops, that means we're not going to have to evaluate all of the combinations, at least if we're mainly interested in that main effect term, that is the general mixing ability. So this is an example from a small study that had 28 peas and 7 barley. So there's 28 peas on the columns, barley's on the rows. That would be 196 combinations. And in this article, what they were able to show is you can get good estimates of the general mixing ability term by just testing 64 combos, with the critical feature being that we have every pea with 2 barley, every barley with 8 pea in this particular example. So the other component, hopefully, that we're going to want to reach for is a genomic relationship matrix to connect the data from multiple environments, which in this case means intercropped partners. So we want to test as many intercrop partners or environments as we possibly can. So the idea here is to use genomic relationships plus this sparse design. Briefly, I'll kind of show you the main figure from the paper we wrote a couple years ago about this. And all I want to show is, so we have this grid here, that's our sparse design. We've got all of the individuals of one species and all of the individuals of another species we might want to evaluate, but we're only going to sparsely test them. So every member of each species gets tested with several members of the alternative species. So the filled tiles there, basically the ones we might select for a field experiment. Then 3 and 4 here, I'm just going to go quickly over 3 and 4 here. Just we're gonna plant out a field trial, collect the relevant phenotypes, whatever they are. We're going to genotype everything, and at the end, we're going to be able to fit a genomic-enabled model of general mixing ability. And this can allow us to do 2 types of selection, the typical 2 that we're interested in, variety development and population improvement. So first of all, we can select all of the best combinations and advance them into some trial where we're actually test those combinations. That would be selection for variety development. And the other part is, of course, improving the population. And this is probably a bit optimistic, but the idea is we're going to run a breeding program, right? And we're going to, through multiple cohorts, make selections, make crosses, and improve or co-improve the system as a whole. So that was the idea. I think the one other thing I wanted to show is we also can simply directly incorporate genomic relationships into this general mixing ability model. So we can simply include a genomic relationship matrix for oats, for the oat GMA, for pea, for the pea GMA, and a Kronecker product of the 2 can help us to capture the interaction term, the specific mixing ability. And importantly, that's going to mean that I can make a prediction of even the untested combinations and their performance, as well as the breeding values of each species, even those that we haven't field tested or that we haven't field tested in combinations. Okay, so that's the idea. With what time I have, I want to tell you about what we're actually doing. So firstly, we have a project that focuses on cover crop mixtures. And we're trying to use both genomic and phenomic selection to do this. The focal system I chose was crimson clover and oat. This is sort of a listing of cover crop species that are used in mixture. So cereal rye is the most common cereal that's used for cover cropping and in mixtures. But a lot of folks where I sit are very interested in grazable cover crops. Oats have a little bit of a higher forage quality. So we chose crimson clover and oat for this project for that reason. And so this is a project that's funded by a USDA NIFA and an Auburn intramural grant. My collaborators include Dr. Esteban Rios, who's a forage breeder at the University of Florida. He has a crimson clover breeding program. I actually met him when he gave a seminar, or remotely when he gave a seminar right here a couple years ago. And we have some partners at Auburn, physiologist Alvaro Sanchez. Yin Bao is now at the University of Delaware. So our new phenomics expert is Tenzil Remen. And I'm very grateful to the Sun Grains Cooperative. Steve Harrison and Ali Babar are small grains breeders in the Southeast, and they have basically donated oat lines to the project. So our major goal with this project is basically just to pilot the the ideas that I just shared, right? Using a sparse design and connectedness from genomic or phenomic information to evaluate these mixing ability terms as early as we can in the breeding pipeline. So before I show you how we're going to integrate this into a crimson clover breeding program, to tell you how we actually breed crimson clovers, they're open pollinated. So this is one of the things I've spent the last couple of years kind of wrapping my head around and figuring out how to make progress. We start with evaluating single plants, right? Space plant evaluation, we do it in a pollen isolated way as much as we can. We plant as far from any other crimson clovers because whatever we let flower, the bees are going to decide what the mating scheme is. Each of our genetic units is basically its own special individual. And so we're going to have to do some kind of culling selection. I'd say 10 to 20% is much more serious than I actually use. But so we're going to allow just the best plants to pollinate. We're going to collect seed from each individual plant so we can track the maternal lineage at least, if not, but we're of course not going to know the pollen parents. Those maternal seed lines then go obviously to the next year's space plant nursery. This is an allogamous annual. We're cycling every single year. And in addition, we can get in the second year our first opportunity to do biomass yield kind of trials. We often call these sword plots, but importantly, each plot is going to have a half Sib entry. So each plant may have the same maternal haplotype, but that's about that's the main connection between them. The other thing that we do with the seeds from each space plant nursery is comprise a synthetic line or a balanced bulk from the best, say, 50 to 65 individuals. We make a single seed lot from each nursery, and we enter that into the Cover Crop Breeding Network's Advanced Line Trial Series. It's a multi-location, like 15 sites up to per year. And this is where we get our first biomass yield and like cover crop performance metrics compared against major commercial checks. This is the test bed where we'll determine what might be valid to release in the future. Okay, so now I can tell you how we're going to integrate genomic selection into this, or specifically genomic selection for mixing ability with oat. So we have our initial space plants, we cull them, we get the selected plants and their seed. It's at this stage that we're going to do genotyping of the half-Sib families. Those half-Sib families are then going to, of course, they're going to be used to comprise next year's nursery to make the next balanced bulk. And the thing that I want to add to this is a yield trial where we can evaluate potentially both sets of genetic units. I've called this a preliminary yield and mixtures trial. But as you can see here, we have a bunch of single row plots, and each one of them has a different crimson clover half-Sib with an oat companion to compete with. Of course, the idea is we can then use the combination of the genotypes on the half-Sibs and the phenotypes that we collect in the yield trial to make genomic prediction and kind of intercept the seed on the way to the next year's space plant nursery to try to increase the intensity of selection and incorporate the prediction for general mixing ability at the least. So the trial is going to be 4 site years total, 2 locations times 2 years. We're actually just finishing up. I will hopefully be harvesting my second and final year of this main experiment next week, in fact. So we have single row plots. Last year they were just 6 feet long. This year we've got 12-foot long plots, better seed yield last year. Other important features of the experimental design, we have around 200 entries per year that we ran, 178 in the first year, 200 this year, including the advanced lines that are currently in CCBN's ALT series. We kept the oat lines consistent since they're inbred lines. It's at least a little more relaxing than having everything cycle every single year. So we kept the 26 oats constant. We ended up with 1,200 plots total, or 600 plots per location year. So just to give you context, instead of going into all the details of how we set up the partially replicated design, there are 4,628 possible combos of the number of germplasm that we had last year. And instead, we've sampled about 22% of that space. So we had 1,020 combos, as you imagine, with 1,200 plots. And as I explained with the sparse design, we're mostly most of them are unreplicated. Some of them are replicated so that we can get better estimates of interaction effects and those kind of things. Right, I'll skip over this part. I also wanted to mention another wrinkle. So this is our first attempt to actually do genotyping for breeding in crimson clover. I mentioned we don't have a reference genome yet. We chose to go with what I call frequency typing or pooling by families. This is an approach that Dr. Rios has used in alfalfa and annual ryegrass, among others, successfully. The idea is we're going to take like equal parts of tissue from 20 plants per family, pool it, isolate the DNA. We used low-pass sequencing from Hudson Alpha Institute's KUFU platform and ended up aligning our reads to the red clover's reference genome for now. So we ended up with 3,700 markers approximately that we can use. And the idea here is we're sequencing to capture allele frequencies and co-frequencies between the families rather than genotypes, right? So this is not plant-level genotyping. But we can construct a genomic relationship matrix from this and use it to make predictions. Also, just to mention that we're looking at a whole bunch of different phenotypic parameters. I'm just going to talk briefly today. We've done a bunch of non-destructive sampling, mainly with multi and hyperspectral UAVs, something like 7 time points per site per year have been flown. And of course, cover crop termination time, we take the biomass, we separate by species, and get the dry biomass per species. So that's our primary yield component and the two phenotypes that I'll talk about today. Well, I have been dabbling in genomic prediction using last year's data set. My graduate student, Gideon Oyebode, first PhD student to join the lab, has been working on the UAV data. So what he's done is he's taken the 7 time points per location. He's taken both the spectral reflectances that come directly from the drone, also vegetation indices like NDVIs, and use that to make as the predictors to make a phenomic relatedness matrix. The intermediate step here is basically spectrally doing spatial correction of the spectral readings and to get blues for each crimson clover family, basically. So I end up with a single value for each of these predictors for every crimson clover family, and we end up with a relationship matrix that's number of clover by number of clover, matches the genomic relatedness matrix. So this is fairly preliminary work, but without further ado, the first thing that I wanted to show is that we have detectable genetic variation for both crimson clover and oat mixing ability from the first year of this field trial. 2 traits are all I'm going to share: total biomass and the proportion of that biomass that's crimson clover. And this is the proportion of variance that's from genetic sources, parsed by the sources specifically. So there's no specific mixing ability. Our trial is definitely set up to target that general mixing ability term. And as you can see, the bulk of the genetic variance appears to come from the clover, and most of that's really captured by that phenomic relatedness matrix. So not much directly from the genomic relationship matrix. But this is a good first step, right? We have statistically significant genetic variance to work with. Next thing I did was to look at those predicted breeding values. And so I kind of wanted to figure out what's going on in the germplasm, start to think about how we would use this for selection. So here's each dot is either a clover or an oat accession, with the x-axis being proportion clover, the y being the total biomass. So basically, we see a significant trade-off, a negative relationship. This makes perfect sense, really, right? Oats that produce a lot more biomass end up corresponding with being less compatible with crimson clovers, basically. Whereas basically we need to be selecting for bigger clovers. So I think this is the hypothesis we would have gone into. Grass legume mixtures, usually the grass is the more competitive member. So we probably need to be breeding for more competitive clovers and less competitive oats, sacrificing some of the total biomass along the way. Okay, so that's one of the things we can look at. And of course, we want to know, can we make genomic predictions with this quantity, with these data? So I'm going to predict two things: the mixture performance, which is going to be mixture total genetic value, in other words, the combination of the predictions for both species and the interaction. And I'm also, of course, interested in the breeding value prediction for the clover, because I'm running a clover breeding program, and that's what I want to use to select what goes to the next nursery. There are different resampling procedures we could do for cross-validation. For brevity, I'm just going to talk about the clover. I'm just going to resample the clovers, but you can also do resampling with the oats. And the hardest thing to do, of course, would be to resample both species at the same time. In other words, test the ability to predict combinations where you've observed neither the clover nor the oat before. But I'm just going to show the clover resampling for today. So we can do some prediction of especially the mixture performance. So shown here in the teal would be the prediction accuracy for the mixture total genetic value. Since we are resampling the clovers here, the oats are essentially, their genomic relationships are always in the model, which means that a lot of this prediction accuracy comes from there. They're basically treated like environments for the clovers. And we've got very small, or let's just say modest, prediction accuracy for the clovers. This is our first attempt, small marker set. And I'll take it. It's greater than 0. So there you go. And as I mentioned, we also have been using the drone data to try to capture relationships between clovers as well. And the cool thing is when you include the phenomic relatedness matrix, that's the teal here, we get much improved ability to predict the clover general mixing ability in particular. So just to kind of conclude this little bit here, I think I have more time. The key things, we do have significant variation for clover and oat performance in mixtures. There's that oat biomass productivity trade-off to contend with. Suggests we need to select for less competitive oats, more competitive clovers. My clover mixing ability prediction is small, but phenomic prediction does very significantly improve that. I do think an open question I need to consider carefully is how I should actually use that, and be happy to hear thoughts about that at the end. And there's lots to do to get better at working with crimson clover and genomic prediction in the species. We probably need a bigger population. We most certainly need bigger plots. We can get them from bigger seed yields. We need to develop the reference genome and get an idea of actual population genetic parameters like effective population size, et cetera. Okay, so that's it for clover and oat mixing ability. I also wanted to spend a few moments and tell you about cassava and cowpea. So in the tropics or in the global south, the slide doesn't specifically show that, but it is a fact that intercropping and polyculture production is much more common than it currently is in temperate systems. So it seems just very obvious that if we're going to breed for intercropping, one of the most important places it's going to be done, it would be in tropical systems. And as you guys know, worked with cassava for a lot of years, and my graduate student Gideon actually worked with the cowpea breeding unit at IITA after he worked firstly for the cassava breeding unit. So when we got to talking about what he would do for his PhD, it was like obvious we should try to see if somebody in our cassava network wanted to plan an intercropping trial with cassava and cowpea. So Gideon set up this trial and pitched it, or we collectively pitched it to Dr. Ismail Rabi, who's now the head of cassava breeding at IITA. And I've just been super thrilled that I've gotten to keep collaborating with him, even if it's just on this small pilot project. He's basically run it for me gratis so far. And I get to share with you the very first year out of 2 that we're going to get to run of this experiment. So this is the experimental design. We have 120 cassava clones that we chose from their existing pipeline, PYT and AYT stage. Gideon chose 2 cowpeas, one that's an upright type and one that's a spreading type. Don't ask me too many questions about cowpeas. Not an expert there. We observed them in both mixture and in monoculture, unlike the previous experiment that I just described, where we only focused on mixtures. And there were 2 replicates with a 2-row design like this. So the cassava and the cowpea are planted in the same row directly with each other. The other thing to know is that they are planted together but not harvested together. It's almost more like a relay, I would say, because cowpeas are short season, 3 months to 4 months maximum before harvest. Cassava takes 10 to 12 months before harvest. So something like 9 months, the cassava are actually alone in the field after the cowpeas come up. I was honestly betting against there being any impact on the cassava at all, but that's actually not what we found. So indeed, when you intercrop cassava, regardless of the type of cowpea that we try, cassava root yields and by various metrics are somewhat decreased. And the same is true for the cowpea. When they're intercropped, they have lower yield than when they're monocropped. Okay, so that's not the same thing as saying that the intercrop itself is not as valuable. In fact, the one result I want to share is this. This is a ranking of the cassava clones, our 120 cassava clones, relative to a parameter called the land equivalent ratio. This is simply the ratio of the yield observed in intercrop to the yield observed in monocrop. And so in the green, we have LER cassava greater than 1, meaning these guys are actually yielding better when we intercrop them than when we monocrop them. And I'm not showing like the details of the mixed models we fit, but we can also show that there's a significant genotype by management interaction here. Both of those collectively say that we have some motivation to breed cassava, or at least to evaluate cassava under intercrop conditions. And we need to do that if we want to figure out which cassava would be the right for intercrop, would be the best for intercropping systems. So I hope we can use this pilot project to sort of launch additional work in this and other kinds of systems. All right, so last but certainly not least, but definitely the most early stage, I have this really cool project with Jean-Luc and others that's focused on a temperate grain intercropping system. So this is a project with oats and peas. Thank you to Peter Hyde and Leah Treffer for this slide. So the main thing that I want to say is, so we plant these are again full mixtures of oats and peas that we plant. The key feature here is if we get maturity correct and we can successfully combine them together, oats and peas have pretty different seed size and shape, so they can be separated after harvest. So basically all I'm going to say today is that we have kind of a breeding for intercropping dream team assembled that includes, again, Jean-Luc, Peter Hyde, and Leah Treffer from here at Cornell. And we have both an oat and a pea breeder on the project, deriving lines directly from their breeding programs. So Juan David Arbeláez may be a familiar name around here, and he is our oat breeder at the University of Illinois Urbana-Champaign. Nono Bendillo started off as our pea breeder. He's now at North Carolina State. Sorry if I got that wrong. So Marisol Berti, who's an expert in intercropping at North Dakota State, is managing the pea breeding program there. And we have a partner at Corteva, both Sarah Lira and Frank Techno, and postdoc Annabelle Laurent. So these folks are, each one of us is planting a single location of this field trial. I actually planted mine in February. I assume that will happen sometime soon in New York. I'm not going to like go over all the different objectives. They're basically, or in some ways, the same as what we were doing with crimson clover and oat. That is, we want to pilot genomic prediction of intercropping ability. And we have this huge latitudinal range, like we're really spanning a lot of environments here. We have a USDA NIFA plant breeding for ag production coordinated innovation network. It's a really mouthful kind of grant name, but this will allow us to do 2 years of a primary field trial series and sort of a supplementary third one as well. We're going until 2028. And again, we're deriving our germplasm for oat and pea directly from middle stage of the breeding pipelines. All told, across the couple of years of the project, we're going to have 480 oats and 440 peas, or somewhere in that ballpark. But each of us is going to plant 400 plots. 10 location years will end up being run total. So that means we'll have 4,000 total intercrop plots, which would mean, with this number of oats and peas anyways, that's 211,000 combinations. So as opposed to the 22% of the space that I searched with crimson clover and oat, we're actually searching less than 2% in this situation. And so I'm really excited about this project. You can see we're just reaching about elbow high, I guess. In my field site, things are growing fast. I actually saw some flowering peas just last week. And so stay tuned for more on this project. And that's where I'll finish. Breeding for intercropping is definitely a major combinatorial challenge. And I'm hoping that the normal things I like to throw at problems, genomic prediction, sparse testing, will help us do early stage evaluation for these quantities. So we need empirical examples, and that's really what I'm up to these days. And with that, thank you guys so much. It's been an honor. Can't wait to hear your questions. All right.
Marnin, yeah, beautiful talk. I was wondering about you have this GMA estimate that's between two species. How correlated do you think that would be to essentially intra-species competition?
How correlated the general mixing ability
The
term?
GMA, yeah, I mean, I'm sure you don't have the answer yet.
Yeah.
What that is, but is competitiveness within two individuals within a species, is it kind of the same process as that GMA process? The SMA, I totally expect to be very different, but I was kind of wondering, do you think that's going to turn out to be 50% correlated? That if you
So this is often called the associate effect or the indirect effect of one species on another. I just for time I couldn't. There's a whole nother like evaluation model where we will separately analyze the separated yields, like just the clover biomass and just the oat biomass. And when you fit that same model with the main effect and the interaction term, the main effect for the alternate, like if I'm analyzing clover biomass, then the main effect for the oat in that model is telling me about the oat impact on the clover. So and then that can be actually added to the GMA for clover to give me, adding to the producer effect for clover. This is a little hard without showing the model, but that's directly actually a measure of the competitive effect versus the so a producer effect would be the species' own effect on its own yield, and the associate effect is the species' effect on the other species. So we can basically break that GMA down into those components, and then think about what to do with that for breeding purposes. And actually, I can say from a little bit of preliminary results that Jean-Luc has shared from some of his work with oats and peas, there is a negative genetic, or is it fair to say, there's a negative genetic correlation to some extent between the producer and associate effect. So yeah, more competitive something, yeah, there's a sacrifice there.
Also, I just want to say awesome talk. So in thinking about like G by E of oat planted by itself, are you seeing anything where G by E would be higher or lower when it's planted with P?
I've just papered over that so far. So you're asking if there's actually less G by E when we have an intercrop versus a monocrop. And we do have some ability to try to estimate those components. I have not done it yet. So what was your question, whether it's a
Higher or lower, but if you haven't, then that's like totally fine. But what would you expect, I guess, then? I'll pose it that way.
I know what I would like.
That is fine too.
I already know from betting against cassava being affected by cowpea intercrop that what I would expect might be wrong. So
we have one question online. Does climate affect pollinators? Does drought, heat affect yield? How is this factored into the way you're sort of setting up your program?
I would say those things are, I mean, relative to the intercropping projects, we have multi-location trials. So we're essentially trying to also estimate the G by E term. I'll take that as a question about like the impact of environmental variance. And I have been trying to focus just on like the genetic component, mostly like main effects rather than those interaction terms. Not sure else.
There was also a follow-up. What
Ah.
are the pollinators for clover. So I think it was geared a little bit more towards
Okay.
pollinators and impacts of climate on pollinators.
So bumblebees and all kinds of other bees love clovers. I'm no expert on like all the specific species of their pollinators. One of my master's students actually is interested in that topic, and so we'll see what she can actually like produce, but I don't have too much knowledge about that. Regarding the environmental effect, one thing that I do think that we'll see when we can dig into like, especially soil fertility, there is when you have a higher nitrogen environment, that really benefits the grass and causes the grass or the cereal to outcompete or to be more competitive against the clover. The clover can fix its own nitrogen, so when you have a lower N environment, and I think that's going to be a major confounding, like in terms of spatial variability, I observe myself like in the first year from one end of the field to the next, we went from more oat, less clover to more clover, less oat. And I can't tell you why because I don't know, but those are definitely confounding factors that we have to kind of address just by spatial corrections, how I've handled it so far.
So I think you highlighted some clear pros that conventional growers would want to have intercrops. Organic producers have the same thing. In fact, they have fewer tools to deploy, to control, and have these benefits. So are you giving any additional consideration in your program to organic producers and what they're looking for in the system?
I'm very, like, I would say, like, especially breeding cover crops, organic producers are going to be some of my best customers or my best customers. Actually, so far, I work on a conventional farm. We don't have an organic farm that I can yet do plant breeding and genetics kind of work on yet. We definitely contend with the issue like planting intercrops, like of oats and peas, for example, immediately excludes the vast majority of any like chemistries that we can use. So there's no post-planting herbicides, for example, by definition. I don't know if that's exactly an answer to your question, but we have the same challenges in that regard.
Actually, I have a very basic question. So when you're deciding a crop or you're thinking about a crop to be a cover crop, what are the key characteristics that it should have, the crop?
Probably a lot of people in this room could answer that better than me. Well, so we're typically, not always, but most of the species we're thinking about are cool season species because most of the commodity production is warm season, right? So it needs to be winter hardy, right? And it also needs to be able to deal with, nobody puts fertilizer or adds, uses herbicides or anything to control things in a cover crop field. The whole idea is, farmers are busy, they're going to harvest as soon as they can, and then they, to plant a cover crop, they have to do extra work. They have to spend extra money, buy the seed, plant the seed, spend the diesel, all that kind of stuff. So the main thing that the cover crop has to be able to do is survive on its own in a low fertility or low kind of care, and it has to compete against weeds. The main three traits we really are breeding for in the cover crop breeding network are biomass, of course, but then early vigor, which basically just means getting big as quickly as possible, so that we have some weed suppressive or erosion control kind of benefits in the early part of the cool season, and then has to survive over the winter. Yeah. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.